407 years ago, our forefathers brought onto this continent a new nation, conceived in liberty. Testing, 1, 2. Testing, check 1, 2, 3. Testing, check 1, 2, 3.
That's what we need.
Hang on, don't start yet.
Well, welcome, and thank you, everyone, for coming out. Scots Science Scholars, you're required to be here, obviously. You are the feature and the highlight today. But for faculty, guests, support staff, thank you all so much for being here and helping us out. So Scots Science Scholars is the brainchild, really, of Dr. Siopsis, and I would say Dr. Brigatti a few years ago, about 8 years ago, when they originally came up with the idea to apply for a grant from the National Science Foundation to fund a program that would provide support for students who are interested in STEM. Dr. Brigatti ended up getting pulled off to work on some other projects, and I can remember the day that Dr. Siopsis came into my office and asked me to jump on board with helping to write this grant, and I of course tried to give her all the excuses of how busy I was and there's no time and it's not going to get funded and no we shouldn't do it, and she of course wouldn't listen to me, and I'm really glad she didn't, and I'm glad she twisted my arm, and we were able to get funding from the National Science Foundation, which is a pretty big deal because not a lot of schools of our size have the grants, this kind of a grant, and we're really proud of it. You guys are our 5th cohort, and according to the National Science Foundation and according to the original grant, this should have been the last cohort. But because of the good work that all of your predecessors have done, and because of all of the great support that we have had from administration here at the college, from staff, so the IT team that comes out and helps us and solves all of our problems constantly, all over campus, from Kathy Pate in transportation, coordinating keys and vehicles to us, to Johnny Freer in the dean's office, helping just keep things organized, Brenda Engel, our division secretary, who keeps us on top of everything and organizes everything. We had tremendous amount of support from faculty and staff. Administration, the dean and the president are very supportive of this program. The Scots Science Scholars have, over the years, and there's some alums here, sitting here, have done good work and made a good reputation for us on campus. Scots Science Scholars is a respected group, and as you remember what Dr. Bogart said on your first day on campus, opening day, he looks forward to Scots Science Scholars coming every year because it's the first group of students to arrive on campus, and I think that we are a good group to be making that first arrival on campus because you're coming to work, and we know you work hard. Y'all are tired, right? You work really hard. But yeah, thanks to all the good work that your predecessors have done, all the people who have supported us along the way and given us help, all of the faculty members who come in and teach you guys, and I know that you'll be giving the shout outs this year to the different faculty members who came in and taught you lessons. To Lindsay Walton, who runs our STEM Success Center and just does an amazing job with motivating, coaching, a little bit of rear end kicking here and there, probably from time to time, keeping people organized, keeping people on task, identifying when people need pep talks, identifying when people need to go to separate rooms to work quietly and study, or when they need to be together in groups. She does an amazing job running that, and Lindsay, we're really grateful. It's added a tremendous amount to our program to have that. We are so proud of you guys, Scots Science Scholars. All of our Scots Science Scholars, all throughout the years, really super proud of you. I can say there's 4 right now that I have to call out. Boomer Russell, who was in charge of transportation and coordinating labs, and great peer mentor for you guys this year. Morgan Gast, who was our record keeper and TGA runner and lab whatever. Morgan Bria in charge of social media, and I know she did a lot of wrangling and bossing folks around. So Bria Jefferson and Tyler Lee, who stepped up originally and said, I'll be willing to be the heavy and make sure everybody gets places on time and make sure people show up on time, and that's a big part of your success, and what you guys have learned from them, I think they are more critical to your success here at Maryville College than anything any of the rest of us have done, because you have seen 4 people who've got it together and who are doing it right. And if you just copy what they're doing, you're gonna be in great shape here. So just do what they do, and yeah, keep it up. So do you have anything else to say, Dr. Siopsis?
Except
anything to
thank
add?
you, and Dr. Gibson was way too, as always, humble about her involvement in this, because the grant would not have been gotten without her. When she came on board, it really brought it all together, and the program has developed over these last 5 years because we're a team. And so I just want to say we're equal partners in this. There's not credit for one of us more than another, and she's my bestie too. So
We're a good team. Y'all know this. So enough of me. We're anxious to hear from you guys and hear about you telling us about all the hard work you've done, all the things you've learned, and hopefully some fun stuff too. So we will start with Julia, Emma, and Daniel talking about the American Eagle Foundation and the trip to Dollywood.
So you need to stay as close to the screen as possible so we can do it.
I'd like to welcome everyone and thank you all for coming to see our presentations today. My name is Julia.
I'm Emma,
Daniel.
and we did our presentation today on the American Eagle Foundation in Dollywood.
So a little bit about us. I'm from Cleveland, Tennessee, and I'm going to be a chemistry major on the pre-med track, and I want to be a pediatrician.
I'm from Lenore City, Tennessee. I'm a biology major with the pre-vet track, and I want to be a veterinarian.
I'm from Wallen, Tennessee, and I want to be a computer science major and be a programmer / computer scientist.
So, S Cubed, as a group, we took a trip to Dollywood on Friday. Dollywood, originally, the land was called Rebel Railroad. It was a single train, and it had a saloon and a general store. 2 companies later, the Hershon brothers bought it and named it Silver Dollar City. And then in 1986, Dolly Parton, who is a country music star, she's known for not only her talents, but her passion in educating East Tennesseans. She partnered with Silver Dollar City, and they renamed it Dollywood. And then in June of 1990, they announced that they had partnered with the American Eagle Foundation and had started an eagle rehabilitation project.
So the American Eagle Foundation has 5 main goals in their company. Their first is to educate people about the different types of birds. They do this by having video cameras in eagles' nests so people can watch safely and learn about their habitats. And they also have outreach programs such as Wings of America at Dollywood that we got to go to, which educates millions of visitors. They also repopulate the eagle species, so they have released 145 eagles into the wild. They typically get 10 to 50 injured birds every year, where they rehabilitate them and release them back into the wild unless they're too injured to go back into the wild when they stay with the American Eagle Foundation for the rest of their life. They also conserve and protect the eagles' nests, and they do this by staying safely away from them and teaching others how to do that too. And lastly, they inspire people to learn about birds and how to protect them.
So all of the birds we will tell you about today are targeted and known for being birds of prey, but only a few of the birds we will talk about, such as the eagles and the hawks, are technically top of their food chain. And the reason that's important is because the ones that are at the top are called indicator species. So when biologists and ecologists get all of their different data, the indicator species are the ones that show the first harmful effects of pesticide and drought and habitat loss. So they're a really, really big deal in getting information for our areas and our species of birds that are here. You will see a lot on our slides, and you will hear us say a lot that all of these birds eat small rodents, and so without them, our rodent population would be crazy. So this is one of the main things that these birds do for us.
So this is the golden eagle. It has golden and brown feathers throughout its body with a yellow and black beak. It tends to live in open mountains, open country, foothills and plains, and in the north and west. It lives in tundras, prairies, and grasslands. It's also located in North America. It tends to hunt in groups, so that way when they're in a tree, when they swoop down to get their prey, so that way when the prey tries to skip the first bird, the second bird grabs it. They tend to eat smaller mammals like rodents and mice, and they sometimes eat foxes and young deer.
The Harris's hawk is a medium-sized bird with dark brown feathers and a white-tipped tail. They are found in North America, typically in New Mexico, Texas, Arizona, and the Colorado River Valley because they like to live in desert areas. They are one of the more social birds of prey, so they are normally found in groups of 3 or more, and they hunt low in the bushes because that is where most of their animals that they eat are found.
So the red-tailed hawk is a broad-bodied bird. It has rounded wings, which differs it from the eagle because the eagle has more straight wings. It's known for its red cinnamon tail. It also has a deep brown back and a tan pale underside. They live in primarily North America, and they prefer the open country because they do spot their prey from high in the air. So if there were a lot of trees around, they wouldn't be able to see exactly what was on the ground. They're monogamous birds, so they mate for life. And both sexes help incubate the eggs. And females are much larger than males, so they're often mistaken for eagles. And then their red tail is seen, and they realize that's not the case. Besides helping with the rodent population, they provide housing for smaller species of birds in the area, specifically swallows, who are known to live in active hawk nests, and they're primarily found in this general area.
So this is the black vulture. They tend to be larger birds, weighing about 3.5 to 6.5 pounds. They have a wingspan of 59 inches and a length of 22 inches. Their habitat is towards the northeast because they've been declining in the southeast. They tend to forage in groups because when they're flying high in the air, they look out for dead animals, but the group in the air, they look out for each other so that way they can locate more food. They benefit to an ecosystem because they eliminate dead carcasses around the area.
The barn owl is a medium-sized bird with light brown feathers and a white heart-shaped face. They are found in North America, and they prefer open land with a few trees and buildings and cliffs that they can build their nests in. They detect their prey using their hearing and low-light vision at night, so they typically hunt for the animals running around the fields at night. And once they catch their prey, they swallow them whole and regurgitate pellets of the undigestible parts of the animal. And then the females make their nests out of these pellets, where they typically lay 4 to 7 eggs.
So this is the bald eagle. They have brown feathers with a white head. They tend to live in coasts, rivers, and large lakes, including swamps in Florida, edges of conifer forests in southeastern Alaska, and desert rivers in Arizona. And in the winter, they tend to move to dry canyons out west. They hunt by cruising over seas and rivers where there are surplus of fish. They wade over the rivers so that way they can spot the fish, and if they can't find any, they tend to steal from other birds. They decrease the overpopulation of rodents in the area.
This is my personal favorite, and I'll tell you why in a minute. This is called the African pied crow. It's actually a raven. It is an 18-inch tall black bird, and it has white on its breast and its collar. It's found in the south of the Sahara, and it prefers open land just like the red-tailed hawk does. And it is in the desert area because that's primarily where most of its insects and lizards live that it eats. They're very social birds, so they're always either in pairs or small groups. And when they hunt in small groups or in large groups, they can actually take down large animals such as calves or lambs. And they're actually known for plucking the eyeballs out of small lambs. So they don't eat them, they just take their eyes, which is gross. When they work together, they can be very, very destructive, and they actually have a global bad rap for being territorial and protective. With the recent climate change in the South, there has been a soar in the population of these birds, and they've actually completely offset the predator to prey balance in their area because they are so destructive on animals that are larger than them.
So the barred owl has dark brown and white feathers, and they have almost completely black eyes. And they are typically found in forest areas, normally in the eastern United States. And interestingly, there have been recent fossils found of barred owls that are at least 1,100 years old, found in Florida and Tennessee. So that shows that they're a very old species that haven't really moved around much. They are more active during the day than barn owls, but they still primarily hunt at night. And they also eat their prey whole and regurgitate pellets, but they don't make nests out of the pellets. They just use the natural cavities found in trees or buildings or cliffs to nest in, where they lay 1 to 5 eggs.
Personally, S-Cube has been an amazing experience for me. I have loved getting to know the professors here and to build a better relationship with them. I was super nervous about getting to know the campus and finding my way around, and I've found myself much more comfortable with the campus and also in myself. I've loved making friends here, and I've loved all the activities and the trips that we have done. And it's been a big wake-up call to me. I knew college was going to be hard, but I wasn't sure it was going to be this challenging, and so I'm very grateful that I've gotten the experience and the knowledge and the help that I have to feel successful.
S-Cube for me has been really great, and I think the best thing for me is that I feel like I got a head start on the other students, because before I hadn't really had a lot of lab experience, but I feel like now I kind of know what I'm doing in the labs, and we just got to go over a lot of things that I was very unsure of, and I got to learn a lot of new things too. And I also love that I got to meet some of the professors and have a relationship with some of them, and I love that I got to meet new people, and I don't have to stress about not having friends on the first day of school. And I love these people, so.
The SQ program is important to me because walking into the program, I didn't know anybody, and now I'm friends with pretty much everybody in the program. I also built a relationship with everybody, including the professors, Dr. Gibson and Dr. Siopsis. And I learned a lot in the past 2 weeks, including in and outside the labs, so that'll give me a head start going in just when school starts.
And we did want to say a special thank you to Dollywood and the American Eagle Foundation for helping us get this knowledge and for inspiring us to do this.
So welcome to our presentation. Today we're going to be talking about computer programming, and this is basically our perspective of the modern computer language. Before we get started, we want to tell you a little bit about ourselves. First off, my name's Hannah. I'm from right here in Maryville, Tennessee. My major is biochemistry, and after I obtain my degree, I hope to be a forensic scientist.
And I'm Justin. I'm from Knoxville, Tennessee. I'm going to be majoring in computer science, and I'm going to be a computer programmer once I get my degree.
So, some common questions about computer programming. First off, what is computer programming? And the answer to that would be, it's basically telling machines what to do through the use of code. Also, another common question is, why is computer programming important? And that would be because it allows people to take online classes, so if they can't commute to their local college, then they can still get their education. Also, computer programming allows us to develop video games and things like PCs. And also, without programming, we would not have cell phones, so we'd probably be using smoke signals or writing emojis on stone tablets. So, another question, what are some applications of computer programming? Programming allows us to do things like build chemotherapy machines and use heart monitors in hospitals. And it really does a lot for us in the day-to-day. Another big aspect of computer programming is computer languages.
Computer languages are custom vocabularies built to help interact with the computer or machine so you can get it to perform a specific task. Computer languages come in low and high level languages. Low level languages are more descriptive, they're more complex, and they give you more control over all the systems of a computer, but they're a lot harder to understand without proper practice using the language. High-level languages are less descriptive, they're easier to use, and they're closer to like human speech, like the code runs pretty much the way you would say that sentence, I guess. Low-level languages include like C++, which is taught worldwide in pretty much all computer science classes. There's also binary, which all other languages have to be converted to binary because that's the only language computers can understand. Otherwise, your program won't work if you don't convert it. Hexadecimal, which is a simplified form of binary, which turns your long strings of ones and zeros into shorter numbers and letters. High-level languages include Java, which is used primarily for making all sorts of different web browsers and a lot of smartphone apps. Python, which is used by a lot of different companies to do prototypes of their hypotheses about their products, because it's really easy and quick to make a little short code to test something out. And then there's also Scratch, which we wouldn't know about without the computer science lab that we had with Mr. Robert Lowe. And Scratch is a very high-level language. It's primarily used for to spark interest in people who are new to computer programming or people who are just amateurs at it. Make them really think about, maybe I want to go to college and get a degree in computer science someday.
While Scratch is used to make creations like games, stories, or animations, it's also got an online community where you can upload your creations and get some feedback and give some feedback and become a better programmer. And speaking of creations, we actually programmed something for you, and we want to show you that.
Me. After what?
Start it again. It's weird. All right, so this is a simple target shooting game we made. You play as the Grim Reaper, and you aim with your mouse cursor. You're trying to shoot. That's not supposed to be on. So you're just trying to shoot the little ghost with your fireballs, and once you hit him 4 times, a little crab will come out on screen. He'll walk around, and he'll tell you that you won. And now we're going to go back to the slides, and we're going to show you how we made this work in Scratch. All right, so this is Scratch coding. A lot of things we're going to see is when the green flag is clicked, that's basically when your program starts. Another thing you'll see a lot is forever loops, which basically means it's going to do everything inside of here forever until your program stops. You're going to see if statements a lot, which is used in pretty much all computer programming. It's if something happens, then do everything inside of it. If that thing doesn't happen, you're going to skip it. So over here with the Grim Reaper that you play as, we're checking if the space bar is pressed, we're going to trigger the fireball, play a sound, and we're going to wait a second. And we wait so you don't shoot like a million fireballs at once. The ghost, we're checking if you're touching the fireball, we're going to add 1 to your score, create another ghost somewhere else, and we're going to make this one go away. The fireball, where once it's triggered by the space bar, the Grim Reaper, it's going to point towards your mouse cursor. It's going to go in that direction, and then if it's touching the edge of the screen, it's going to go away. The crab, it's checking if your score equals 4, which you can change it depending on how long you want to play. The crab's going to show up, it's going to walk across the screen, and it's going to tell you that you won. None of that would have been possible without the computer science lab we had with Mr. Robert Lowe through the S-Cubed program, which is why we want to talk about what S-Cubed means to us.
So S-Cubed has really expanded my horizons as a biochemistry major because before this program, I hadn't really considered the other areas of STEM. My plan was to come in here and do the school, graduate, move on with my career, not pay attention to STEM at all, basically. But S-Cubed has really exposed me to the other areas of STEM, including the computer science lab, which actually led me to find out that I love computer programming and am now considering it as a minor. Additionally, S-Cubed, while they've done fun things like kayaking, going to Dollywood, and ORNL, they've also given us plenty of chances to partake in math labs and science labs and teach us those key lab procedures we're going to need. Overall, S-Cubed and its leaders have prepared us not only for our upcoming STEM courses, but also our futures in STEM as well.
S-Cubed for me, as well as broadening my horizons, making me think about STEM in ways I usually wouldn't have thought about before. It's had a much larger impact on my social life. Because before this program, I would come home every day from school, I'd lock myself in my room, I'd start playing video games. I wouldn't really talk to anyone, I wouldn't really talk to my parents, I'd only ever talk to my dog, really. But during this program, I've spent a lot more time out of my dorm. I've been hanging out with friends, we've been doing all these bonding and trusting exercises through like mountain challenge and stuff like that. We've really gotten to know each other, and I've really come to find a lot of people that I can rely on to help me through the next couple of years at Maryville when things start getting a little more tough. So we want to give thanks to all these people.
And especially Mr. Lowe for taking time out of his day to teach us the computer science lab. No, that's fine. Sorry. Yeah. You want to talk about Piglo Lab? Where did that go?
There we go.
My bad.
Here. Yeah.
Hi everyone and welcome. I'm Colby
I'm Megan.
I'm Lauren.
and we did our presentation on Mountain Challenge. So a little bit about ourselves. I'm from Port St. Joe, Florida. I'm majoring in biochemistry with a pre-med route, and I want to be a cardiac surgeon.
I was born in Baltimore, Maryland. I'm currently majoring in biology, and right now I'm undecided.
I'm from Sparta, Tennessee. My major is mathematics, and I want to be a math teacher. So a little bit about what we're going to tell you today. We're going to tell you about Mountain Challenge and why it's so important to Maryville College specifically. And then we're going to tell you how closely Mountain Challenge and STEM are related, because their director does a very good job at incorporating them. And then we're going to tell you what STEM and S-Cubed has meant to us this summer. So Mountain Challenge was founded by Bruce Gilliam, that's him up there, and he wanted to show people that you can not only be physically fit but mentally fit as well. And the way he does this is through his program. It's based towards incoming college freshmen, and they want to show them how to use different social skills with their peers, and they do this through activities that require you to trust them and use teamwork. But he also does a very good job at incorporating academic into it too. And they use the motto Fit, Green, Happy because they want you to be physically fit and healthy. They want you to be environmentally aware and protect your surroundings. And they want you to be happy. And we couldn't find a picture of the logo, but we did find Dr. Gibson wearing the shirt, so she's going to model it for us.
All right, Mountain Challenge relates a lot to STEM. For one, it takes outdoor activities and combines them with critical thinking. It kind of forces people into a group of strangers that they've never met before, and it basically forces them to talk to each other, and it helps you build social skills. It also applies math, science, and engineering skills. The engineering can be seen in the way that the certain challenges are built and the way that he incorporates them. And you have to use math and science within the challenges, and a lot of science and engineering can be seen in the belay system, the Crawford House, and the Maryville College Woods.
All right, so Crawford House is where Mountain Challenge runs out of. Crawford House was tested through Gold Means Leadership in Energy and Environmental Design, and on this test you can get a possible 110 points. Crawford House holds a 66, and Maryville College is one of only 3 in Tennessee colleges to have this type of facility on campus. And when they're tested, they look at things such as your electricity, water, waste. And so one thing Crawford House does to cut down and get that high score is they use solar power to help cut down on their electrical cost.
All right, the Maryville College Woods, this covers over half of our campus. It's It's 140 acres, and there's a 100-year plan that protects the woods and everything that's in it. A lot of students use this for their senior thesis because there's a lot of nature and different components. A lot of research and new discoveries are made in the woods every day, and it's primarily a sanctuary for wild animals such as the red fox, the northern slimy salamander, the barred owl, and the eastern box turtle. However, there are a lot of invasive species, primarily plants, such as the English ivy, the Chinese privet, and the bush honeysuckle. And as you can see on the left, that's the English ivy, and on the right, that's the bush honeysuckle.
On our first official day, we all went to Mountain Challenge and we did 6 activities. We did the multitasking and tennis ball game, and the wall, the spider web, the island, and the zigzag balance challenges. So here are 3 of the activities we did. First one was multitasking game. So what we did is we went in this big giant circle and we each had an object. And when Bruce, the leader of Mountain Challenge, when he said right, we had to pass the object to the person to our right, and when he said left, we had to pass the object to our left. That was easy. We messed up a few times, but then he made it really, really hard. So he told a story, and the same thing. When he said right in the story, we had to pass to right, and when he said left, we had to pass to left. But then in between sentences, he would say a math problem. So it'd be like, what's 7 times 7, or what's 4 plus 8, and we would have to answer that while passing. And so we failed a lot because it got really difficult and we could not multitask. The wall, the objective for the wall is to get everyone over it, but once you're over it, you cannot help lift someone up. What we had to do, we had to use engineering to build like a foundation. So we used strong and big men to bring everyone up. And we used a football player, Cordell, and we used Jose. He's a wrestler. And so they lift everyone up. But then we realized who's going to lift them up. So then we had to change our foundation completely. The only people left was 3 girls and 1 boy. So as 3 girls, we were actually confident. We were ready to lift these big men up, 250 pounds. But Jose, he had no faith in us whatsoever. He was not ready to have us lift him up. We did fail our first try. We dropped him just a little bit. But he didn't hit the ground. We had him, but we did eventually get him over, and we completed the challenge that way. And then the island, my group did not do that, but Colby's did, so he will explain it.
All right, so the island, there are 3 pallets set up and they're evenly spaced out. However, you can't get to each island unless you use the 2 boards that are given. You have a long board and you have a short board, and you must lay them across each island, and you have to use simple physics such as leverage to make it across. There has to be 6 feet on the board at all times, and only 4 people can fit on the second island. So you have to keep passing the boards back and forth in order to get everybody to the 3rd island. Yeah, we failed a lot and had a bunch of harsh punishments, but we eventually made it to the 3rd island. And that's that.
So here's a whole bunch of pictures from the first day. As you can see, this is Colby placing down a wooden plank to another island. And over here is the circle where we had to be passing objects. And this picture right here is Jose figuring out that 3 girls are going to have to lift him over the wall. So as you can see, his face is like, no.
All right, so this is the Alpine Tower. It's 60 feet tall, and it's all run by a belay system. The belay system includes a carabiner, a rope, a belayer, and an ATC. The most important component of the belay system is the carabiner. It's a simple hook that hooks the belayer to the device, and it's made out of the same metal that airplanes are made out of. That way it's primarily indestructible. That way if there's too much tension on it, it doesn't break. And when you put tension on the rope, it puts leverage between the carabiner and the ATC. That way you can lift the climber or you can drop the climber, but it keeps them from hitting the ground. And it's all run by a pulley system, and without engineering, it wouldn't be possible.
So here's day 2, and we got to climb the tower. I was really excited for this. No one else was really excited, but it's okay. So I found it really easy climbing it because I was excited. I wasn't scared. But then when I got to the top, I realized I had to get down. And that's the part that freaked me out, because I had to trust my partners, Julia and Emma, so that they wouldn't drop me. Because he said it's 60 feet in the air, and you just kind of have to jump, and I didn't want to fall. But after I trusted them, they did good. They didn't let me die, but I enjoyed it.
I'm gonna be honest with you. I was really scared to do the tower. Like, I just wanted to roll up in a ball and start crying. I got 20 feet up and I just wanted to come down. But all of the peer mentors and everybody else kept saying one more step, and I realized that I had to stop having self-doubt and to build confidence in order to make it to the top of the tower. And once I got to the top, I kept grabbing the rock at the top and I couldn't pull myself up. And I just wanted to give up, and everybody was like, one more step. And finally, I pulled myself up to the top, and that symbolizes a lot of how college and your college experience can be, because if you get up, you can't go as far as you thought you could in life. So you always have to go the extra mile.
The tower for me, I was ready to go up, and then I got up there and realized this was not what I was expecting. And one thing that Colby said, the one more step, Bruce said that to us. He said before we got up, he goes, when you're ready to stop, we're gonna say, take one more step, give us one more step. And so what he wants you to learn from that is you're always gonna reach your limit in life, and you're gonna have to take that one extra step, because if you don't, you're never gonna reach your full potential.
So what does SQ mean to me? To me, it means opportunity and family. So opportunity is labs. Like, labs is completely different from high school. And so I'm glad I got to experience that without actually having to deal with a grade. So I got to do it before then. But with family, everyone's so easy to talk to. Everyone was so nice, and we just all talk. Like, it will be 1 o'clock, and we'll all be down like the common room talking. And it's so crazy. And once you think like someone's gonna say, okay, I'm gonna go to bed. Instead, Boomer, he was like, you guys wanna go to cookout? So we all went to cookout. And that's just insane, because back home my bedtime's 10:30, and it's just like I'm out at 1 o'clock in the morning. So I really enjoyed it. These 3 weeks have been the best part of my summer.
S3, I just want to say thank you for this wonderful opportunity. Without this, I wouldn't have the confidence to do what I've set my mind to do. I've been given a lot of experience in the different STEM fields. I've had a lot of lab experience, because in high school I didn't get really much lab experience. Now I do. And it's allowed me to grow as a person and to find myself and to better myself. And I've made friends that I'll probably be friends with for the rest of my life. And as you can tell, the first day we didn't really want to touch each other. We were like, ugh. And on the last day, that was the baseball game 3 days ago, and you can tell that we all like each other, and we know that I think we like each other, but we're gonna be friends for the rest of our life.
Asking for me, I was very scared coming up here. I was afraid I wasn't gonna be able to do anything right, and I wasn't gonna make any friends. But then you get here, and immediately on the first day, we're very uncomfortable with each other, but we're still talking. And then you get thrown into Mountain Challenge with Bruce, and he makes you just immediately trust each other. He's like, you're gonna have to be together and work together. And so that's been great, because you're making friends that you're gonna keep for the rest of your life. But while you're doing this, you're getting so much college experience. And one thing I love about this place is they do live up to their, when they say that your professors and everyone here wants to help you as much as they can, that's the truth. Every professor we've worked with has been overly helpful, and it's been the greatest thing ever. And we're getting experience that we never would have gotten had we not come here.
We just want to give a big thank you to everyone that was involved with STEM, but we want to give a huge thank you to Bruce Gilliam and his Mountain Challenge crew, because without them, none of this would have been possible.
All right, hello everybody. My name's Liam,
My name's Cordell.
and we decided to do the P-Glow lab with Dr. Gibson as our presentation.
All right, so a little bit about us. Again, I'm Cordell. I'm from a small town called Tiptonville, Tennessee. I play football here at Maryville College. A couple of things I like to do, hunting and just any type of athletics, and then I plan on majoring in engineering.
And I'm Liam, and I'm from Nashville, Tennessee, and I've competed in a couple of different sports in high school, but I will currently be playing football this year for Maryville College. My hobbies include playing the PS4 and astronomy, and my major is going to be biology.
So, what does S cubed mean to us? Really, it's been a great experience with a bunch of great people here. I've not met anyone I don't like yet. It gives us a head start on campus life and a head start with all of our classes. There's lots of learning with all STEM fields, so you get a broader view of STEM.
All right, so the purpose of this lab that we did is to genetically modify the bacterium E. coli to make it fluorescent. Scientists genetically modify organisms all the time to benefit society, and we just did sort of a low, sort of like really simplified version of like showing like the processes of how to do things in the lab, as well as genetically modifying bacteria.
Some terms to know: a genome, an organism's complete set of DNA, including all of its genes. Ampicillin, a semi-synthetic form of penicillin. GFP, green fluorescent protein. Arabinose, a sugar that is used to induce the protein of GFP. And then Luria broth, a medium used to grow bacteria.
Okay, so a question that we were asked by Dr. Gibson before we started this lab is she's asked us, why do you think we're using bacteria to do this experiment? And so our answer is we feel that bacteria have a high production rate, so that means that we have a lot of results to look at, and they the production rate's very high, so the results come quickly. So this took just about an hour to get all of this bacteria to actually glow. The bacteria are one of the easiest subjects to control for an experiment, so you don't have to worry about, a live mouse maybe biting you or escaping in like a cage or anything like that. All you have to worry about is contaminating the outside environment. So basically, we just had to wash our hands when we were done. And the third reason is single-cell organisms are easier to use for genetic transformation. It's just a simple organism. I don't have to worry about, with like a complex organism with all the cells and having to make sure all of those cells are transferring information. It's just one cell that is dividing into bigger cells, or into more cells. Okay, so the way that we make bacteria glow is with plasmid. And you may ask, what is a plasmid? A plasmid is a chromosomal ring that's found in bacteria. This ring is replicated by bacteria automatically, and it contains genetic information for ampicillin resistance and for producing green fluorescent protein. Scientists commonly use plasmids for cloning genes and modifying organisms, or in this case, scientists have genetically created the P-Glow plasmid, which is a plasmid that we use to transfer the information for the E. coli to actually glow, and it's made up of 4 different parts. It's made up of arabinose, green fluorescent protein, which we actually get from jellyfish, beta-lactamase, which is a protein that is on all the time, and it provides resistance to ampicillin, the antibody ampicillin, and it has Ori, which stands for origin, which basically makes this entire plasmid replicate its genes.
Okay, so some uses for the green fluorescent protein. It's commonly used in medicine and research to study gene expression and protein function. Glowfish have been genetically modified with the green fluorescent protein, as you can see up in the top right. Scientists can use GFP to track gene expression in cells or in the body. All right, so the lab procedure that we had, first we had to add the plasmid to the bacteria, so separate from the plus or minus control. And then we had to heat shock it and then cool the bacteria. we had to add the bacteria to the agar plates, and then we had to grow the bacteria in an incubator for about an hour. And then once that was done, we took it out and inspected the bacteria.
Okay, so what is happening while we're doing this is called genetic modification. And so E. coli is naturally killed by an antibiotic, ampicillin, that we've been mentioning. And ampicillin destroys the bacteria's cell wall. I don't know if y'all have noticed this, but E. coli is in your poop, and your poop doesn't glow. And the plasmid P-glow encodes two important proteins, beta-lactamase and GFP. And so what happens is when we insert P-glow into the E. coli, the beta-lactamase degrades the ampicillin, and then the GFP makes the bacteria glow. So the beta-lactamase is basically kind of like the protector, and the GFP is the thing that makes it do its job.
Alright, so here's what we got to expect from this experiment. Over there in the top left, we have no DNA on these agar plates. With Luria broth, we expect grow but no glow. With Luria broth and ampicillin and no DNA, we expect no grow and no glow. So then we add DNA with Luria broth and ampicillin, we expect grow but no glow. And then finally, when we add the DNA, we have Luria broth, ampicillin, and arabinose into it, we get grow and we get glow.
And then this is just pictures from the experiment while we're doing it. Here you can see Morgan working with Jose and Daniel, and then here's actually where we heat shot the bacteria, where we basically just took the bacteria and put it in some heated water at, I think it was 52 degrees for a certain amount of time, and then we take it out. And then this is just me and Cordell actually adding the actual bacteria colony to our agar plate right here. And then Dr. Gibson helping out Lauren, I think, in that picture. And then our results from the experiment. We have all genetically modified bacteria. Everyone in this room that is part of the SQ program has genetically modified bacteria. That's pretty cool, right?
Hey, Dr. Gibson, very happy.
Yeah, this is a big part of the results right here is Dr. Gibson. But this is our actual agar plate that we, with the bacteria that we actually colonized and successfully inserted the plasmid P-glow into the bacterium E. coli that made it do what you're seeing it do right here.
Alright, so then here's just a chart of the results. And everything happened as we expected it to. So when we added the DNA, we had with Luria broth and ampicillin, we had grow but no glow. And then finally, when we added the DNA and had the Luria broth, ampicillin, and arabinose, we had grow and glow. And then with no DNA, Luria broth, ampicillin, no grow, no glow. No DNA, just Luria broth, grow but no glow.
And when we say DNA, we're referring to the plasmid P-glow itself.
And then we would just like to say a big thank you to everyone on this list, but especially Dr. Gibson for helping us out.
Dr. Gibson, and then also a student that did this last year, Cameron, helped us out with this slide a lot, so we need to give a special shout out to him as well.
Mathematical models used to predict it.
So my name is Dylan Banks, but I go by Felix. I'm from a small community about 20 minutes northwest of Asheville, North Carolina called Leicester. I'm pursuing a computer science and Spanish degree in hopes to obtain my master's degree after Maryville. My favorite parts out of this SQ program were the team building and the bonding exercises. They were my favorite because they allowed me to feel like I was actually contributing something for once in my life. The SQ program showed me many different areas of STEM besides just computer science that I want to do, such as chemistry and biology, and without the program, I don't believe I'd ever have experienced them.
My name is Mae Nicole, and I'm from a small town in northwestern Pennsylvania called Cambridge Springs. As of right now, I want to major in pre-vet, and my favorite part of the SQ program was the opportunity to explore the different STEM fields offered here at Maryville College.
My name is Cooper Reed. I'm from Spanish Fork, Alabama, and I'm majoring in biology, and my favorite part of this program has been the lab experience that we were able to have.
Our initial purpose was to determine the number of species on an island at any given time. In order to do so, we needed to apply mathematical models to estimate the immigration rate of all species to a previously unpopulated island from the mainland, as well as the extinction rate of all species already on the island. Mathematical modeling itself consists of equations derived from calculus and other fields of math. These equations, called differential equations, are applied to real-life situations in order to solve complex issues.
Now, you might not have guessed this, but in a mathematical modeling lab, we do have to do a little bit of math. So let's start with something called the discrete difference equation, which the basic formula for that is n of t plus 1 equals a function of n of t. So basically, let's say n of 2 equals a function of n of 1. So to know the next number in the series, you need to know the previous one. So obviously, n0 has to be given beforehand, and equilibrium is reached when nt plus 1 equals nt. So what you put in, you get the same number. And before we do all of our calculations to apply this to the lab, we need to make some assumptions. The two factors we're using are immigration and extinction. So let's assume that those are linear functions with opposing slopes. So extinction has a positive slope and immigration has a negative slope. And
sorry.
So if we'll just use R as number of species and T as time, we get our basic formula of RT plus 1 equals RT plus immigration minus extinction. So I'll just change that immigration and extinction to functions of RT to keep this kind of theme we have going. Right, so let's plot the function of extinction. So as you can see here, we have P and E of x and the point 0, 0. In this graph, P equals the maximum number of species that can be on the island, and E represents the maximum number of extinctions. And since you have maximum number of extinctions when all the species are on the island, then we have the points E, P, X, and 0, 0. And if we just use the slope formula, we can kind of find the equation of this line. If we do the same thing for immigration, then we can get the function for that as well. And now we just take those two functions, plug them into our equation, and we can kind of get started, right? Not exactly, because the EX and IX, max extinction, max immigration, we aren't really sure what those are yet, but we can figure that out through experimental procedure.
After performing the necessary calculations to define our models, we then simulated the immigration and extinction patterns of the species on the island. The simulation consisted of 200 popsicle sticks, which represented the individuals of all the species, as well as 2 islands of differing sizes, which were represented by pieces of paper. Every 20 popsicle sticks were labeled with different letters to show the different species, and 20 to 21 trials were run for each island in order to show an extended period of time. For each trial, popsicle sticks were thrown at the island for 15-second intervals. This first image here shows the island before a trial was run, and this image shows the island after a trial was run. This popsicle stick here represents an individual of species B. It was outcompeted by this individual of species O, but since there's another individual of species B elsewhere on the island, it does not qualify as an extinction. This individual of species J is the only one of its kind on the island, so when it was outcompeted, it did qualify as an extinction. This individual in the upper right corner landed more than halfway off the island, meaning that it didn't actually make it there and most likely died along the way. This individual of species S is new to the island, but species S was already on the island previous to the trial, so it doesn't qualify as an immigration. These individuals of species O and species M are the first of their kind on the island, so their arrivals qualify as immigrations.
So we got a little more math. So we collected 3 main pieces of data. We collected the number of species, which is represented by variable R in the second column here and the first column here. The number of immigrations represented by variable I, which is located in the third column and the second column here. And the number of extinctions, which is represented in the fourth column and the third column here. Taking the number of species, we plotted those on a scatter plot on the x-axis and then took the number of immigrations and plotted them on the y-axis of same scatter plot. After doing that, we found the best fit line, which allowed us to find the I of x, which was really great because we needed that. Doing the same thing for the extinctions, we took the number of species represented by variable r, plotted them on the x-axis of another scatter plot, took the extinction, plotted them on the y-axis of the same scatter plot, forced it through the y-intercept so that it would look correctly and do what we needed it to do, and it gave us the E of x of p. Doing that, it allowed us to then plug them all back into our difference equation model, which gave us these values. After doing that, we plugged them onto a line graph with the time being on the x-axis and the number of species r being on the y-axis. After plugging that, we showed that the simulation can converge until a point right, if y'all can see, about 8.3, which gives us r of EQ, which is the equilibrium point, which is when extinctions and immigration cancel each other out. And you can be seen through all 4 of our graphs: a small island at 1 meter, a small island at 2 meters, a large island at 1 meter, and then a large island at the 2nd meter.
So further expanding on the graphs that were created and explained by Felix, we were able to conclude that large close islands experience the highest rate of immigration, and large far islands experience the lowest rate of extinction. We were also able to determine that the minimum immigration was 0 and occurred when all possible species were on the island, and the minimum extinction was also 0 and occurred when all possible species were on the mainland.
So everybody take a big deep breath. There's no more math. So SQ meant to us kind of generalized a lot of things. It allowed us more STEM exposure. Like I had said, I'm a computer science major, and I thought I was going to come and just do computer science, just nerd out really hard on that. But like the PGLO lab I thought that was like probably the best lab we've done, because biology school and genetic modifications are bomb. And so it also allowed us college prep. We've been here for 3 weeks, a little longer than everybody else, so we kind of have an idea of where we're going. Kind of. I got lost twice. It also allowed us to forge bonds with each other and also with some of the professors. Big shout out to y'all. Appreciate that greatly. And it also allowed us for career explorations such as ORNL and the TBA. It allowed us to show basically where we can go past college and not just be stuck in one place.
We would like to thank all of these nice people on the slide, but especially Dr. Siopsis, our math mom.
Yeah, right there. So, what it is, yeah.
Having it. This is not it. It's spinning.
Please hold.
This is getting me back. Wait for a second. A little bit.
Who's someone?
You go ahead and introduce yourselves. You remember that part.
I do remember that part. I just didn't know if y'all wanted it with the picture.
Okay,
You have the real life.
we probably should just hold on because until that's. Little tiny
Oh man.
It's gonna be fine. It's my fault. I think I
The
know.
best for the last, guys.
I was so proud of this. I was like, I'm discriminating.
It was good all up until this point.
Yeah.
Good job, Zach.
It's a side.
Woo! It felt like 5 minutes, but it was really 30 seconds. So.
All right.
Okay. So first, I would once again like to thank you all for being here and for watching our presentations. But before we begin our presentation, we would like to tell you a little bit about ourselves. My name is Mae Davis. I am from Gulfport, Mississippi. Right now I plan to do a major in neuroscience on the pre-med track.
Hello, everybody. My name is Jose Antonio Galanchongo. I'm from Knoxville, Tennessee, and I plan to major in biology on the pre-med track as well.
Hi, my name is Susanna Maley Warren, but I do go by Maley, so don't call me Susanna. Unless you work here, then you're good. I'm from Dublin, Georgia, and I also plan on doing biology on the pre-med track.
Before I get into what is a solar eclipse, I know that we all know we saw one of the most incredible natural phenomena, which was a total solar eclipse on Monday. Before that eclipse even happened, Ms. Gierno and Dr. Tolliver, with 3 Maryville College students, 2 of which who were previous Scots science scholars, were working with NASA to collect data for the eclipse. They were gracious enough to let us be a part of that. So we'll get to more of that later, but right now, I'm gonna tell you what exactly is a solar eclipse. Well, a solar eclipse is whenever the moon comes in between the sun and the earth, casting a shadow over the sun. There are 3 different types: a partial, a total, and an annular. A partial solar eclipse is whenever the moon only casts a shadow onto part of the sun, and it is the most common type of solar eclipse. A total solar eclipse is what we experienced on Monday, and is whenever the moon covers the sun completely. An annular solar eclipse is whenever the moon appears farthest away from the sun, causing it to look like a small disk over the sun.
During totality, there are 3 major things that we can see. One is corona, diamond ring effect, and Bailey's beads. And for me, on Monday, my favorite one was diamond ring effect. What about you?
Oh, it was so cool. But the diamond ring effect was really especially cool.
So you may ask, why is an eclipse so significant? And this eclipse was very significant because it was a total eclipse. Most eclipses that happen, we cannot see because it happens on open waters. And this was the first eclipse that came through the United States. Last one was in 1979, and it came through northwestern states.
Throughout history, scientists have been researching eclipses and how the solar eclipses affect humans and their surroundings. Now, I don't know about you guys, but Monday's solar eclipse made me want to run laps, and I'm not a runner, so there's that. Speaking of Monday, on Monday we were able to see, we were able to see a 360 sunset, which looks like that if it was a panorama, and also shadow beams, which look like that. It's kind of like, if you can't see the picture very clearly, it's kind of like the bottom of a pool, where like the water makes that that thing effect with light. It's cool. The next projected total eclipse in the U.S. will be 2024, and it will range from Texas to Maine.
This eclipse not only affects humans and the environment, it also affects animals. For example, cows returning to barns, birds roosting, and cicadas changing their songs.
Last week we went into a lab with Miss Gierno. Thank you for all your work. I didn't know what a UVA UVB ray was before this lab. It's UVA and UVB stands for ultraviolet A and ultraviolet B. They're sun rays, and UVA is only slightly different from UVB. It goes down all the way down to your dermis, which is the deepest layer of your skin. UVB only goes down to the epidermis, and UVB causes like sunburn, sun cancer, the works.
So in Ms. Gierno's lab, we were able to work with veneer sensors and probes. We personally worked with the UVA and UVB sensors, and we split up into different teams and went outside and took calculations on the UVA rays before the sensor was covered, and then the UVA rays once the sensor was covered with a shirt. On the graph here, we have the UVA intensity on the y-axis, and then we have 2 bar graphs. What we did with the data from the sensors is we took one UVA calculation every second for about 63 seconds, and then average those calculations together. And we did that for both without the shirt and with the shirt. Whenever we did it without the shirt, this is our average UVA rays that we calculated. And then the orange-yellow graph is our average UVAs that we calculated with the shirt on. As you can see, wearing the shirt significantly decreases UVA rays that reach the sensor, which also means it decreases the UVA rays that reach your skin. This white bar right there is our standard deviation, which shows the varying UVA rays calculations that we got.
Can you click on the video, please?
I think I can. It's just, I think I know what I have to click.
Well, this video shows the eclipse as it goes through the United States. As you can see, this is the sun, and it shows how the moon affects the sun. This black dot over here is the moon, and the fading shows that as it gets lighter, it becomes more partial than total, and this is the path of totality, as you can see.
As Ever said earlier, when we worked in our lab with Ms. Guillermo, and also while we were taking data on Monday, we used vernier sensors. We used an anemometer, which measures wind speed with in meters per second. We measured temperature. We measured light intensity with a light sensor. We also used a barometer, which measures barometric pressure, which is kind of like atmospheric pressure.
Some other sensors that we used were UVA and UVB sensors and a radiation monitor. However, you will not see our wind speed or our radiation monitor graphs in this presentation.
This is the UVA rays data that we got from Monday. We did this approximately around 12:50. As you can see, the y-axis is the UVA, and the x-axis is the contact 1, 2, 3, and 4. I want to point out some few things like these dots over here. That's when the sensor was blocked by either a person or a cloud, and dots like over here, it's when it was moved and it was pointed towards the sun. And I want to point out something else. On contact 2 and contact 3, we can see a flat line. That's when totality hits, and it lasted around 97 seconds. Thank you.
As I said earlier, UVA and UVB are very similar, so our graphs are very similar. The peaks are, the y-axis measures UVB, the x-axis measures time. The peaks are when the sensors were in direct sunlight, the drops are when they were obscured by like people walking by and stuff. This is a flat line where where totality happened. You might notice a slight gap in between our data and the graph. The sensors were not this sensor wasn't calibrated.
This is our light intensity graph. We started this one also at about 12:50. You can tell in this first section it is not very light intense. That is because it was in the shaded area. But you can see the flat line in between C2 and C3, which is second and third contact. And that is whenever totality happened. Right after totality, you can see a sharp increase in light intensity, which is whenever it started moving into the C4 fourth contact area, which means that the moon started moving away from the sun, allowing more light to come through. And on the y-axis, we do have the light intensity, and then the x is the time. This is our temperature graph. I had 2 temperature sensors attached to my computer, and they took data at the same time on the same graph so we could compare those 2 sensors very closely. You may notice that they have a little bit of a gap in between them, and that is because I had them on different sides of a metal rack, so they're not completely the same. On the y-axis, we do have the temperature, and on the x, once again, the time. And these sensors were started about 20 minutes before the other sensors that you just saw, and that is why this dip where totality happened in between C2 and C3 is approximately 20 minutes after the 100, whenever the other graphs fell outlined at 100.
This is our barometric pressure data. On the y-axis, Edward, could you point out? There's only a slight variation in the intervals. It goes up by 200ths of a kilopascal. On the x-axis, that's of course the time. It does look like there's a major decline in barometric pressure, but as I said, it's only 200ths of a kilopascal.
So what S cubed means to us. Before I came here, I was homeschooled for high school, so I did not have access to any real lab material or anything like that. So I was very nervous coming in as a STEM major, knowing I'm going to be in all these labs with all this material that I've never had to work with before. So being able to be part of the S Cubed program and coming in, they have taught me proper lab etiquette and how to use all these instruments I did not even know existed. And it's been so beneficial. I feel so much more prepared for going into these labs.
All right, for S-Cube for me, it was like an opportunity to experience college life because I was nervous. I was like, what am I going to expect doing this? And it turned out great. It also showed me that I have to step up my game from my high school level to my college level if I want to succeed. And also, I bonded with everybody in here, and I consider them family now.
I love S-Cubed. It's great. As Ever said, I'm really glad for the lab experience, and as I said, I'm very glad for the college experience and getting to meet all these amazing people that I now call my friends. My favorite part was our field trips. We went to Dollywood, which is really big for me because I've never been to Dollywood. Who knew? I also really like the Smokies game, and of course ORNL. I'm gonna call myself out. I've never really heard of ORNL, which is Oak Ridge National Laboratories. So when we went there, it was way cool. I was in awe of all the scientific instruments that I didn't know what to do with them. So yeah, and also of course the Eclipse. The Eclipse is so cool. What an experience.
So the last thing that we want to leave you with is thank you. Thank you to Dr. Siopsis and Dr. Gibson for all that you've done for this program and for making it possible. Thank you to especially Ms. Gierno and Dr. Tolliver, as well as Cameron, Kathleen, and Seidel, who are the Maryville students that worked with them. But it was really incredible for you to let us be a part of that research and let us, just kind of jump in whenever it was their thing.
We also want to thank our peer mentors like Bree, Morgan, Boomer, Tyler, and also Cameron, even though he's not our peer mentors. Y'all are great. Y'all made this 3 weeks awesome, and I just want to thank y'all.
We also want to thank, of course, our featured guests throughout our experience. It really means a lot to just get to hear you guys talk about your passions in STEM and get to teach us what you're passionate about, of course. And we, of course, want to thank the lovely Miss Lindsay Walton, our SSC director, our godsend, our angel, our mom away from home.
Thank you so much for being here, particularly our busy getting ready for the new semester faculty who are here. That's really helpful. Thank you again to Eric for letting us live stream this. And guys, you did this. This was the last thing you had to do for the summer program, right?
Yeah.
Think about how much you grew from that first day. I have watched you grow just in these 3 weeks, and I'm really looking forward to watching you grow for the next 4 years. And guess what? In a week, the real work starts. So we will be there with you when that happens, and we're looking forward to it. So give yourselves a round of applause.